I'm Daniel Levine, and this is the Bio Report.
Though structure-based drug discovery has long been used, technologic advances have given this approach greater power. ShouTi believes its next-generation computational structure-based drug discovery platform will allow it to develop small molecule drugs that are superior in safety and efficacy to biologic and peptide therapies. We spoke to Raymond Stevens, CEO of ShouTi, about the company's structure-based drug discovery platform, how it works, and why he believes it will be able to produce small molecule drugs that will be superior to biologics. Ray, thanks for joining us.
Thank you, Daniel, so much. I'm excited to be here.
We're going to talk about ShouTi, its recent launch of Basecamp Bio as its discovery engine, and its efforts to develop small molecule therapies to replace biologics and peptides. I think most listeners will understand the benefits of small molecule drugs compared to biologics, but if you think about global health problems, these differences become amplified. Can you offer the case for why small molecule drugs are desirable?
Yeah, absolutely. For me, I think there's three primary reasons. One, as a chemist, I'm always amazed at what small molecules can do to reduce human suffering and to treat various diseases. And as a chemist, I have to say, I think whatever a biologics medicine can do, why can't a small molecule do it better and make a better medicine? The second reason is, and this is sort of personal, very early in my academic career, as a professor at the Scripps Research Institute, my daughter was 4 years old, and she came down with a rare disease called Kawasaki's disease. This is where the immune system decides that the circulatory system is foreign and basically attacks it. She was treated with a breakthrough biologic, Remicade, and she was saved by that drug. So I'm incredibly grateful to the breakthrough biologics that really have transformed healthcare. But during the whole process, one of the things that stayed in my mind was, we're so lucky to have access to these medicines. What about children, around the world that don't have access, to these biologics medicines? And then third, as a family with 3 kids, we have traveled around the world, and I've seen firsthand in the world that, more than half the world does not have access to these great biologics. Even in my home state of Maine, some in the state don't have access to biologics, certain types of biologics. There's a really big, I think, unmet need for more accessible medicines. And there's a technology solution, small molecules. Now, my investor friends also point out, Ray, it's great that that's what gets you up in the morning and has you so motivated. But, you also need to, make these things so that, it is a business at the end of the day. You have to support it, which is correct. And that's where it comes full circle. I think small molecules can be best in class to treat different diseases. So those are the reasons why I think, small molecule drugs are so desirable globally.
You say if you can do it with biologics, why not with small molecules? But I take it there are challenges in terms of going after specific target. What's been the limitations of small molecule drugs, and do you think there are limits to them, or do you believe we can hit virtually any target with a small molecule?
Yeah. So first, Daniel, I want to reiterate, biologics have revolutionized healthcare. And it's really been over the last 20 years. We've just seen so many great breakthroughs. And there's 3 primary reasons, I think, of why biologics are so good. One is I think there's safety, speed, and less risk. Biologics are great at validating a drug target in the body. We now know, with so many cases that they've proven to be quite safe. You can get them to patients and to the market very efficiently. This allows one to go through the clinic faster, increased chance of success, relatively sort of safer. However, I also think that there's been a big breakthrough in safety access for small molecules over the past, decade plus. We have better assays to, tease out, any type of safety risks that might happen. So, at ShouTi, we see a real opportunity to go after biologics validated targets. So, we just mentioned, you mentioned this in your question, these biologics, they validate the target. They demonstrate that this target is a good target to go after to treat a certain unmet need or disease. And so once it's been validated, going after it with a small molecule, it allows, in a sense, de-risks it. And so we have a reduced risk on biology, and we can still fill a really big unmet need globally.
ShouTi is using a structural approach to drug discovery. When we hear about platform technologies these days, it usually involves AI. Structural approaches have been around for a long time. How have emerging technologies changed structural approaches and what you're able to do with them?
Yeah, so I've been doing, biotech and also been an academic professor for 25 years. And I've lived through what I would call three waves of computational drug discovery, really dating back to sort of the 1990s. And these computational companies, they generate fantastic tools for us, whether it's computational methods to design a molecule into a binding site, to visualize it. And I think we're now seeing what I refer to as sort of the third wave of this, with AI and other machine learning techniques. So, they're great breakthroughs. They contribute to the field. But I personally do not believe today the computer can do it all. I heard the same things 25 years ago. I heard the same thing 15 years ago, and I'm hearing it now. At the end of the day, you have to make the molecule. You have to understand the chemistry and the biology of the molecules in the body. And this is something that still requires great drug hunter skills. And so what I would sort of complement to your question, AI is helping us. It's a tool, but it's not going to answer everything. So one of the approaches that we've been adding on to this AI and machine learning are techniques such as cryo-electron microscopy. We and others have integrated it with machine learning. And when you combine the experimental techniques with the computational techniques, it's incredibly powerful. Just as a quick representative example, in cryo-electron microscopy, we can visualize and we can see at atomic detail the protein molecule, the drug target. We then use facial recognition technology, machine learning, to see all those protein molecules and exactly what they look like. And then we can bring them together and find similar looking molecules, and we can see the shapes and how the shapes change. And so it's really, to me, about integrating the methods, not just AI alone. And then lastly, I would say what's most important, you really got to integrate, both AI and machine learning with the chemistry, with the biology, and with the predictive methods for safety that we talked about before. It's really I think the magic has always been, how do you integrate all these great tools together to make better and safer medicines?
ShouTi talks about not only replacing biologics and peptides with small molecules, but using small molecules to be more effective. How precisely can you change a molecule to alter its properties to enhance it?
Yeah, so one of the, to me, the first step, in developing a small molecule is, we look at binding. We look at the interaction. I always use the lock and key analogy, where the small molecule or the medicine is the key and it fits into the lock in the body. So we can see that detail. The exact shape we can see. Binding, the interaction, I think, is only part of what makes a great small molecule. In fact, 10, even 15 years ago, we knew how to design the interactions so we could really optimize binding, how potent the molecule would be. So I think that's become pretty straightforward, relatively straightforward to do, improving the binding affinity. What's still really challenging is, the PK and PD properties, the bioavailability. What will the molecule do when it enters the human body? It's amazing still today, as we go through animal trials and we try to model these things. What exactly is going to happen when a molecule enters the body? That's still where it takes, I think, a great drug hunter, that intuition. It's all about balancing all the different properties and needs. And so that's something that we try to do. I think the field has learned. And at ShouTi, we really focus at the very beginning on how do we balance all the properties, not just potency, not just binding, but all the other PK, PD, and bioavailability properties.
ShouTi is initially focused on targeting G protein coupled receptors, or GPCRs. What makes this a compelling target for you?
G protein coupled receptors, they account for approximately 1/3 of all marketed medicines. It's a well-established protein family. The other family a lot of times people think of are kinases or proteases. GPCRs are the largest. We have in our bodies, based on the Human Genome Project, we have 826 different G protein coupled receptors. You see because of a GPCR rhodopsin. You smell because of olfactory receptors, GPCRs. Our hearts are pumping. We have molecules like adrenaline that control certain functions. And so they're clearly, it's a really important drug class. The reason why I went into the field when I started as an assistant professor at UC Berkeley 25 years ago, I really wanted to get structural information for G protein coupled receptors because it was kind of considered to be one of the holy grails of the field. One is a membrane protein that made it particularly challenging, but two, because they are so important for our body function, as we were just discussing, I thought they were really important to structurally characterize these. And finally, in 2007, we finally had a breakthrough with the first human G protein coupled receptor structure. And once we got that, we developed the process to get a lot others. And now we have structures of, I think, more than 150 different GPCRs that control functions in our body. And so those are now available for structure-based drug discovery.
How difficult a target is that for a small molecule?
Most GPCRs, the fact is they can be screened with traditional high throughput screening. And that's why we've actually already today, GPCR accounts for 1/3 of all marketed drugs. So it's, again, clearly a very druggable target. But only, 75% of GPCRs have not been drugged or not with a small molecule. And that's a lot of opportunity. So many of our body functions currently are not targeted yet. And so, in part, one of the examples of which, type of receptor GPCR has been most challenging, what we call the multi-domain GPCRs, class Bs, something like, GLP-1 receptor, where they have two domains. Their natural ligand is a peptide that, in a sense, stabilizes these different domains together. It's been more challenging to make a small molecule for these types. But again, with the recent breakthroughs in GPCR structural biology, combined with cryo-EM and machine learning, combined with the better safety tools, great new opportunities emerge today.
Now, what's it about your approach that gives you confidence you'll be successful at going after targets that have traditionally been hit with biologics? My sort of, initial answer to that question is, first, it's experience. The old saying that investors invest in the team first, it's true. And we have a great, drug discovery team with great small molecule drug hunters. So I think that's one aspect. Again, I've been doing this for 25 years. The other members of the team have a lot of experience, really focused on small molecule drug discovery. Second, getting the structural data, as a previous question that you asked, previously we didn't have easy access to this structural data, particularly in real time.
And so getting access to this type of data in real time really gives us an advantage to move very fast and efficiently. Our cost of target to IND is less than half the industry average. And so, we're quite pleased. So far to date, the company's been going for a couple of years, but we've been quite efficient at going after these. And then the one follow-up point to this is, as I mentioned earlier, we don't like to take risks in biology. There's just a lot of opportunity to convert, these biologics validated targets and convert them to small molecules so that they can be more accessible to all, but also be best-in-class medicines that can be taken orally. So they're better patient compliance. They don't have the storage, refrigeration needs that, biologics often do. They can be shipped and stored for long periods of time. So, a lot of different reasons there why, we believe why we have been successful already.
Take me through your drug discovery process. How do you go from a target to a drug candidate?
We probably spend most of our time, and like most companies, we spend most of our time on target selection. Where you start, that's where that is one of the most important decisions. I mentor a lot of students, former students that start companies, and they quickly have a target and they just want to use it as proof of principle. And I always keep reiterating to them, your choice of target, that's where the momentum starts, and it's really hard to let go once you start investing into that. So we analyze the targets very carefully based on our company mission, which is to make best-in-class medicines accessible to all. That's where we really want to make a difference in the world and what gets us up every single morning. So a lot of time is spent on target selection. But once this target is selected, back to your direct question about how do we go from to the drug candidate, we created a special entity called Basecamp Bio that focuses on the creative process of early drug discovery, and then we integrate the preclinical with clinical very early, probably earlier than most, to make safety a key component in the design process.
Your lead candidate is in phase 1 testing for pulmonary arterial hypertension and idiopathic pulmonary fibrosis. Why did you start there?
Yeah, and the third area where we started was also metabolic and diabetes. When we were starting the company, and we knew that we had the power of this platform of taking a structure-based approach, converting biologics, validated targets into small molecules, we asked ourselves, where is the biggest impact that we can make globally? Where's the biggest global unmet need? And again, like the target choice, we had a good long debate about this. We chose chronic diseases for two reasons. First, we thought it was the biggest global need. Pulmonary, cardiovascular, and metabolic are, three of the biggest areas that affect, the planet. Second, it's also where do you not focus? I have a lot of friends. There's so many great minds that are focused on in areas like cancer. We really didn't think we could add, a lot of value there today. So, but the overwhelming, part of the answer was, chronic diseases. This does, by the way, create a big challenge for us because most biotechs do not go after chronic diseases. Most, the investors, get intimidated by the large phase 3 trial sizes. And so it takes a certain, amount of, commitment to go after chronic diseases, as well as a certain commitment from, specific type of investors that, want to go into this because they see the global big picture for this. Back to, the immediate question of, pulmonary arterial hypertension and IPF, it was really, we were focused on pulmonary and metabolic and cardiovascular. PAH is a disease that there's clearly a big unmet need, particularly for remodeling. There's a lot of vasodilators out there that work really, really well that can help you with the symptoms of the disease. But there was a hypothesis where there was a peptide molecule, a peptide drug that, had proof of concept in the clinic. We looked at it and said we could make a small molecule that could replace this that would be much better, have much better PK and PD properties. And so that's the reason, for all those reasons, the Venn diagram sort of centered on this particular disease.
How are these conditions treated today, and how effective are existing therapies?
Yeah, so as I mentioned, most people, when you find out, first of all, just identifying pulmonary arterial hypertension is a long process. You go to the cardiologist, they send you over to the pulmonologist, and eventually you find out that you have PAH. The main way that you're treated is with vasodilators. And again, these help with the symptoms of the disease, but they really don't alter the progression of the disease. And so trying to have a drug, a medicine that can help both treat the symptoms and the remodeling or anti-remodeling, this is really, really important. So I think the whole area, there's a new generation of medicines that are starting to come out. You may have seen the TGF-beta, Merck acquired Acceleron, and there's some other medicines that are now starting to come out, but they're not in the market yet. This is, for this disease, there's a really big unmet need to, not just treat the symptoms, but also treat the disease itself. So that's where we're heading with relative to existing therapies.
What's your candidate, and how does it work?
Yeah, so the apelin receptor is a receptor that, again, it's obviously a G protein coupled receptor. And as a G protein coupled receptor, the natural endogenous ligand is called apelin. And there's another ligand called alabella. These peptides, hormones in your body, they express both in the pulmonary as well as the cardiovascular systems. And so it's been demonstrated that they can improve cardiac output while at the same time start to help with the remodeling in the pulmonary system.
You mentioned you're also working on a therapy for type 2 diabetes. What's your candidate for that, and how does that work?
Yeah, so, kind of the holy grail of GPCRs, particularly from a structure-based drug discovery, is the GLP-1 receptor. And there are right now peptide drugs that are out there, and they work, quite well. Most of these are injectables, and so patient compliance can be an issue, as well as cost. We believe that with a small molecule replacement of those, there'll be one, as some of our KOLs tell us, my patient, my diabetes patient, they want to, get in the car, drink their coffee, and take their pill and drive to work. And we also believe that the pharmaceutics properties, the bioavailability, the PK/PD can, better be controlled through a small molecule. So again, it's a validated target, very well established. Pharmaceutical companies have done beautiful work over the years, really demonstrating and understanding this particular receptor. And so we're able to follow that path that they've really blazed and try to come up with a really good small molecule replacement.
So this would, what existing therapy would it replace?
So there are GLP-1 receptor agonists that are on the market right now. You've probably seen the commercials on TV, Ozempic, Rybelsus, Trulicity. And so these that are already on the market, these are the medicines that we'd be looking to at least complement what's currently out there.
Last month, you unveiled Basecamp Bio. This is a wholly owned subsidiary that will fuse ShouTi's pipeline and also partner with other pharmaceutical companies for high-value targets. What was the thinking in setting up the subsidiary?
So over the years, I've learned a lot of lessons about what's worked well, and also what have been some of the challenges in building biotech companies. One of my personal frustrations is, I've been very fortunate, I've started a number of companies, but they get acquired. And when they get acquired, that's great. That's great for shareholders. It's great for the employees. It's great for the patients as well, because when we partner, we're partnering or we're selling the company because a pharmaceutical partner can help us to get the medicine, to the market, very quickly, very efficiently. But when that happens, most of the company winds up getting kind of disbanded. One of the recent companies, I started a company called Receptos in 2009, focused on G protein coupled receptors and structure-based drug discovery. The company was acquired by Celgene. They wanted the S1P1 agonist asset. So that was great. It was a great success story. That medicine is now on the market, sold by BMS, called Zeposia, really helping MS patients tremendously. But the learning is, it was unfortunate. Everything else that we had done, the team that we had built, all the other assets, really, weren't of interest, but there was a lot of value still left there. So we decided when we set ShouTi up, we would use the subsidiary model. And so each of our programs are in a Delaware C corporation. So if we do want to partner or acquisition, we can do it very efficiently and effectively. And then back to the immediate question, Basecamp Bio, one of the other experiences that I've seen in companies that I've been involved in, as well as other companies, is that as soon as a company goes into the clinic and times get tough, the first thing that gets cut is often early stage discovery. And I didn't want to see that happen this time. And we had a unique opportunity to recruit an entire team, all at once, where we could focus the early discovery. We would keep it as a separate, wholly owned subsidiary. That way they could focus on early technology development, early drug discovery development, kind of like a Skunks Works project from day one, and maintain that culture of creativity very early on, or at the same time enforcing, and I shouldn't say enforcing, but really strengthening the process of preclinical and clinical and getting everything through efficiently, getting these molecules to the clinic. So that's why we created Basecamp Bio.
Raymond Stevens, CEO of ShouTi. Ray, thanks so much for your time today.
Daniel, thank you so much. I really appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.